Jdaviz is an open source Python package to visualize and analyze JWST data. And it is built for JWST, but it can work with many different observations from other instruments from other observatories. It is a single package that includes 5 preset configurations. So we have Imviz, that is for images, SpecViz for 1D spectra, SpecViz 2D, that has 2 viewers, one for 2D spectra and one for 1D spectra, so you can extract interactively your 2D spectra to 1D spectra, Cubeviz for 3D data cubes, and MOSViz, which is our little beast that is for visualizing many 2D and 1D spectra as a quick look tool. It can be used as a standalone application, just like you imagine other data analysis tools that are around for JWST. So you install it and it's an app and you open it and you just interact with it. The big strength of Jdaviz is that it can work also in a Jupyter notebook. So you can start your own workflow, your own analysis, then you visualize the data in Jdaviz and you can do things by clicking or you can command the interface with code from the notebook, and then you can get things out of the interface and continue your analysis in the notebook. And third way to use it is by embedding it into a web page, and this is what the archive does. So MAST allows you to quick look data in Jdaviz directly in the MAST web page without installing anything on your machine. So what can we do with Jdaviz? There are a lot of use cases that we can do. They're generally inspired by the scientists here at the Space Telescope Science Institute, but they're inspired by the community too. So if you have suggestions, if you have thoughts on how you would use Jdaviz, let us know. And you can contact us with the help desk or with a GitHub issue if you're comfortable with that. Reach out to us and tell us how you would use the tool and give it a try and see if there is something missing from the workflow you would like to do. So I'll show you 3 example workflow now, and then you'll see the actual demonstrations later by my colleagues. So one very basic workflow that can be completed with Jdaviz is when you work with images and you want to look at multiple images of the same field, maybe taken a deterred position so they're not all on the same pixel grid. So what you can do is you load the images and then you can align them by work coordinate system. So the objects will be aligned on in the same place in the interface, so that when you have all the images loaded and you have them linked, then you can blink between the images and see the same patch of sky in different filters, for example. Another workflow is with 3D cubes. So you can load a cube into Cubeviz, and then you might want to look at parts of these cubes. So the spectrum from the center of this object. I highlighted a few blue pixels in there, and the blue spectrum in the spectrum viewer is the sum of the signal from those pixels. And then this particular object has an outflow that goes from left to right, or from right to left. I don't remember anymore, but I can highlight the pixels on the right-hand side, the red ones, and those on the left-hand side, the green ones, and see the spectra in those regions of the cube. And you can see how different the features are when looking at these different spectra. Another use case could be you have many spectra in your sample and you want to extract them from 2D spectra to 1D spectra, and you have a pipeline to do so. So it's all automatic, but then you know that some failed extraction. So what you can do is you can load the failing extraction, the spectrum that is failing in SpecViz 2D, and then you can extract manually the spectrum with the interactive using the interactive bits and pieces of SpecViz 2D. So you see where your trace is, where your background is, and what kind of extraction you want to use, what algorithm you want to use, and you get a sense of how your extracted spectrum changes when changing the components or the methods to do the extraction. So these are examples. There are many more. But to get you started, you'll need to know where is everything. So Jdaviz lives on GitHub. All these are links, and these slides are shared with you. So feel free to explore these links. Jdaviz lives on GitHub. As I said, it's an open source package. The documentation is on Read the Docs. We have example notebooks on GitHub that you can download and use to try the various workflows for all the configurations. And there are also some example videos if you prefer to follow a video instead of reading through the documentation. And we're working on adding more of these videos to the documentation. And so the last thing, let's discover the interface. And you can use this as a reference when you play with Jdaviz or during the next presentations. So this is an example of Imviz. All of the configuration have a very similar configuration of menus. So I'm giving you the basics with Imviz, but you should find your way with the other configurations as well. And so the data menu is to the left, and you open the data menu, and this is where you find what data sets you've loaded. You can unload and reload or delete data sets from here. The zoom settings are in the top bar to the right, and there are two ways of zooming, through a box zoom or with scrolling. So you'll see two ways. You can create subsets and select regions, just like I showed for Cubeviz, but you can do it everywhere. And this is the Create Subset Regions tool that is in the bar as well, next to the zoom. When you explore an image or a cube or whatever, or a spectrum with your mouse, you'll see the coordinates and the values in the top panel. So those are the info from the mouse location. To the top right, you'll find the link to open the documentation. That's always useful. It will open the documentation in a new tab in your browser. You can find the logger communication. So if you do something in the tool and you don't know exactly if it went well or not, and you miss a message from the tool, you can open the logger and you'll find all the messages that the interface throw at you during your workflow. Some are red if it's a problem, some are orange if it's a warning, or some are green if everything went well. And the last one is the button to open the data analysis and visualization plugins. So basically to interact with your image and to do things like modeling or extracting measurements or anything, you'll find all of it in the plugin tray that is to the right. And when you click it again, it will close the tray and you'll have more real estate for your image.